All right, we'll go ahead and get started. Good morning, guys. I'm Gaurang. I'm one of the cardiology fellows here. And good morning, and thank you for attending our grand rounds. Today's grand round is a part of the Congdon leadership. In 2010, Natalie and Jack Congdon established the Natalie and Jack Congdon Visiting Scholar for Cardiology, which enables VCU Pauley Heart Center to attract a number of talented cardiologists. I'm pleased to introduce to you our speaker, Dr. Rosenzweig. Dr. Rosenzweig graduated from Harvard College, where he majored in biochemistry and molecular biology. He received his medical degree from Harvard Medical School and completed his residency in internal medicine and fellowship in cardiovascular medicine at the Massachusetts General Hospital. He is the inaugural director for Institute of the Brain and Heart at the University of Michigan Medical Center. He has previously served as the chief of cardiology and the co-director at the Congdon Heart Center and the director of cardiovascular research at Mass Gen Hospital. His research has focused on identifying novel mechanisms and therapeutic targets in heart failure using the exercised heart to understand what keeps the heart healthy. Dr. Rosenzweig has particularly been interested in transitional potentials and these discoveries and is the scientific co-founder of a biotech startup focused on developing scientific therapies for long QT syndrome. We are pleased to offer both CME and MOC credits for a Grand Round conference series. To claim the credits for today's session, text the code as shown at the beginning and the end of the presentation to 804-625-4041. For questions, log on to cmecloud.com or contact Shannon Winston. I'll now turn it over to Dr. Rosenzweig. Thank you.
Thank you very much, Dr. Prabhu. I appreciate the kind introduction, and thanks to Fadi and colleagues for the invitation. It's great to be able to share some of our work with you all. I apologize for not being there in person. Hopefully, there'll be future opportunities for that. So I'm going to try and share my screen. You may quickly see the limits of my technological inadequacies, but let's see if we get this to work. All right, does that look okay, or?
Looks great.
You're seeing one slide, great. Well, thanks so much. And as Dr. Prabhu mentioned, we've been quite interested in seeing if we could supplement our understanding of disease by trying to understand what pathways keep the heart healthy. And I'm going to try and make the case this morning that there's much that we can learn from that may have value in thinking about novel clinical therapies, particularly for heart failure. These are my conflicts of interest. As mentioned, I'm a co-founder of a recently funded biotech startup that is looking at small molecule therapies for long QT, and I consult for some companies. Several of those listed there are companies I'm trying to convince to take forward clinical trials for some of the targets we'll talk about. And my former employer, MGH, has filed patent protection on some of these targets and approaches on which I'm a co-inventor. So I know it's not really needed in this audience, but I did want to start with a little bit of clinical context. I think everyone here recognizes that we've made enormous progress in the treatment and prevention of cardiovascular disease. On the left is a graph from a perspective article written by Betsy Nabel and Eugene Brownwald for the bicentennial of the New England Journal of Medicine, in which they point out the declining rate of death for cardiovascular disease. And I think all of you know that if you come into the emergency room with a heart attack now as compared to 1950, when this graph starts, your chance of dying has dropped about 75 or 80%, which is really remarkable when you think about it. On the other hand, on the right side, you see a graph from the American Heart Association that is current and projected costs, total costs of cardiovascular disease. And I know the numbers are too small to see, but it goes out to 2030, and it estimates that the total cost in the United States alone will be something on the order of $800 billion. Obviously, the financial burden of cardiovascular disease is only one metric that reflects the human burden of this disease, but it's shocking to think about how much of a burden this is on our society. So how do we understand these or reconcile these two views? Why is it that we've made so much progress, and yet all of you know we're confronted with really an increasing burden of cardiovascular disease? And I really think there are three principal drivers of this. First of all, in some way, it is a somewhat paradoxical burden of our success. So we have gotten really good at getting people through those heart attacks. In 1950, we just watched, before my time, but people just watched folks have heart attacks, and bed rest was the primary therapy, which had a host of adverse consequences, as we'll talk about. And now, it's an imperative, obviously, to open the artery and reperfuse as quickly as possible. And those things have really contributed to the declining death rate from these acute insults. On the other hand, the patients who survive those heart attacks go on and are at risk for developing the late sequelae of their ischemic heart disease, particularly arrhythmia and heart failure, which we are less good so far at definitively treating. The second issue is that, as all of you know, we are in the midst of an epidemic, not only in this country, but throughout much of the world, of cardiometabolic disease. And so that is a driver, has been long recognized as a driver of vascular disease, but I just want to emphasize that it also drives all forms of heart disease, including heart muscle disease, heart failure in particular, and arrhythmia. And then finally, our population in the United States and throughout much of the world is aging rapidly. And so the people over the age of 65 in the United States are the fastest growing subset of our population. And all of these diseases are closely linked, particularly heart failure, to aging populations. So I think these three drivers of an increased burden of disease are somewhat the explanation for why, despite the progress we've made, we're still confronting an epidemic of cardiovascular disease, which, as you all know, remains the #1 killer in the United States and throughout much of the world. And so not only is it still a problem, but the nature of the heart disease we're seeing is changing. And so when I was a fellow, we saw a fair amount of uncomplicated, single vessel disease that we treated. And I can't remember the last time our cath lab saw a, simple angioplasty. It seems like all the cases are complicated, both by comorbidities and complex disease. And in particular, as mentioned, we're seeing an increase in the amount of heart failure that we see. And in particular, among the heart failure with preserved ejection fraction, or HFpEF population, is what is particularly increasing, as shown in the graph on the right. And this is probably because that's the form of heart failure that is most closely linked to both obesity, metabolic disease, and aging. On the left is an old slide showing survival data after an index hospitalization for either HFrEF or HFpEF, heart failure with reduced or preserved ejection fraction. At the time, I think it was surprising to most clinicians, including myself, that the prognosis for HFpEF was just as bad as the prognosis for HFrEF, acknowledging that these are different populations with different comorbidities and age demographics. But nonetheless, surprising in that regard. It has improved since then, but still overall, after an index hospitalization, is probably around 50%, which is on the order of some bad malignancies. And I think sort of underappreciated. So in this context, as you all know, we have a handful of classes of drugs that have been proven to improve the hard clinical outcomes of patients with HFrEF, with heart failure with reduced ejection fraction, but really much more limited repertoire of drugs that seem helpful in HFpEF. We can improve how people feel, and some drugs like SGLT2 inhibitors have been approved, but really on the basis of reducing hospitalizations, which is important, but I would argue not as dramatic as we would hope in terms of improving clinical outcomes. And so, there's a need for new therapeutic approaches. And in that context, 7 years ago, we became interested in the idea that exercise seems to not only prevent, but mitigate these diseases, and wondered if we could learn something from that. Of course, in humans, there are a lot of confounders in terms of who chooses to exercise and how much they exercise. And it's a little hard to parse out what's causal and what's not. In animals, we can actually study those things more rigorously. And so some years ago, in an age-related model of heart failure with preserved ejection fraction, Jason Rowe, when he was in the lab, showed that not only does this, as is well documented, prevent heart disease, but in his study, he showed that it could actually reverse many of the phenotypes, not all, but many of the phenotypes associated with heart failure with preserved ejection fraction. When he did RNA sequencing, what he identified as the top 3 or 4 pathways associated with this were all associated with cell cycle progression, which I will come back to, but none of the pathways previously linked to HFrEF, heart failure with reduced ejection fraction, showed up as being important in this context, again, an animal model, but still perhaps reinforcing the observation that most of the drugs, almost all of which that are useful for HFrEF have been tried now in HFpEF and have failed to meet their primary clinical endpoint. So some years ago, we became interested in sorting out whether we could identify the pathways responsible for the benefits of exercise, whether some of those pathways were in fact intrinsic to the heart, because obviously exercise does a host of beneficial things. It changes your metabolism, it changes your skeletal muscle, it changes your systemic vasculature. But we wondered whether there were also intrinsic pathways in the heart that were contributing to these benefits. And if there were, could those be leveraged somehow to identify new targets and hopefully new medicines specifically for heart failure. So again, hard to do this in people because of all the confounders and also because of the lack of access in tissue to heart tissue, thankfully, most of the time. So we studied this in animal models, and we started out with a swimming model shown here. This was a very high-tech swimming pool that we had for the mice, which honestly was a tub we got from Target for about $4.98. You have to watch the temperature. You can see there's a thermometer there. Mice are not natural swimmers, so you have to have a good lifeguard. And Nina Mann was the medical student at the time, now an assistant professor at Harvard Medical School, who was supervising these mice. She'll rescue one of the poor swimmers in a few minutes. And we would train them sort of 10 minutes twice a day, increasing that time so that by the end of about 2 weeks, you see she's helping one out there, they were swimming about 90 minutes twice a day. So 3 hours of swimming a day. So, and candidly, the reason we did this is I thought when we started out, we would need to do something this dramatic to see changes. In fact, that's not the case. And so almost everything I'll show you, we've now transitioned to mostly use the voluntary wheel running model. Mice are not natural swimmers, as I mentioned, but they are natural runners. So mice run, if you just give them wheels, they're nocturnal, they will run 5 to 7 kilometers a night, which is quite a bit for a little mouse. I find this somewhat infuriating because that's about what I run, and looking at them, I'm pretty sure they're quicker than I am. Just so that's a little bit upsetting for me. But the nice thing is they will run a lot, and everything I'll show you really we see in the running as well as the swimming. So you don't need to do such an intense model. And also, the swimming model I think is appropriately criticized because it is stressful for the animals. They're not natural swimmers, and so potentially confounded by that. And also, if you're not careful and don't have a good lifeguard like Nina Mann, the swimming model can easily turn into a drowning model, which is an interesting question, but not what we're interested in. So, and then what we did was to use a bunch of kind of modern high throughput omic technologies to try to identify what are the pathways that are differentially regulated in exercised hearts, and what can we learn about that. Honestly, when we started, I thought that we would see in both, and compared that often, the exercise models to pathological models, for example, aortic constriction, where we create a dramatic increase in afterload, and the hearts grow in both conditions, both the exercise hearts and in response to increased afterload, as you know. And honestly, when we started, I thought we would find a common set of pathways that were altered in both of these models, and then a few that were unique. In fact, that's not what we saw. So I think the three most salient lessons that we learned from these are, first of all, that almost nothing is in common between these two states when you look at the molecular pathways. And again, very surprising to me. I thought most of it would be in common. One of the recurring themes of my talk and lab is that I'm often wrong. So this was an example where I was wrong. And in fact, when you find the pathways, molecules, and we've done this at RNA, non-coding RNA, coding RNA, protein, post-translational modifications. It's a recurring theme that we see in all of these profiling approaches. When you identify things that are statistically changed in both models, most of the time, there are a couple of exceptions, but most of the time, they change in opposite directions. So to me, and this is even at really early stages when the hearts look identical. So I'm not talking about late when the pathologic model has transitioned to heart failure, when obviously things would be different, but even just a couple of weeks into the models, these things are quite different. And so to me, that suggests that really from an early stage, the exercise model sends the hearts down on a very different, at least mechanistic pathway, which could be interesting, and kind of speaks to an old debate, which has been, is exercise really different, or is it just a dose effect? So even if you're Michael Phelps and you exercise, 6 hours a day, you're still resting 18 hours a day. And so one of the hypotheses has been that maybe the difference between exercise or physiologic growth of the heart and pathologic is really just a question of degree. If you have aortic stenosis, it's obviously there 24 hours a day, 7 days a week. Whereas if you exercise, it's intermittent, and maybe that recovery is what makes the difference. But this would argue a little bit against that, really early on, before there are any pathological signs, the pathways that are being modified are actually quite different. The second theme that has recurred is that in virtually every case, in truth, every case that I'm aware of, when you look at the pathways that are functionally important in the heart's response to exercise, those pathways also protect against pathologic stress and either prevent or reverse, in some cases, disease. And that's interesting, and obviously was part of our goal, is to see if we could identify therapeutic targets, but didn't have to be that way. It could have been that exercise was just beneficial but independent and didn't really do anything then in pathologic models. And I'll show you some of those data. So these two observations or themes together suggest that if you study exercise models, you'll come up with a different list of candidates, a different list of pathways, and that those pathways are enriched for functionally important and potential therapeutic targets. So that's part of what kept us interested in this. And then the third observation, which was not part of our a priori hypotheses, was that when we looked, as I mentioned with Jason's paper in HFpEF, very consistently exercise activated pathways that were linked to cell cycle progression and regeneration. Not so much in the heart, which, as you know, has a very limited capacity to grow new cardiomyocytes in the adult heart, but in other systems. And so we wondered whether, in fact, exercise could be driving a regenerative response in the heart. As probably most of you know, it's harder than you might think to figure that out. So part of the challenge is that cardiomyocytes, the main working cell in the heart, often have more than one nucleus. And so you can have a cell that is synthesizing DNA, but it's not really undergoing cell division. It's just making a second nucleus, or it's creating more than the normal amount of DNA. And then the other issue is that a lot of the techniques that are used to look for markers of proliferation are in fact non-quantitative. And so even if a cell just repairs a little bit of DNA, it'll incorporate things like EDU or BRDU and turn positive. But again, it doesn't prove that a cell's undergoing cell division. So some years ago, a colleague at the Brigham, Rich Lee, had a beautiful paper in Nature documenting a new way to analyze these situations and really get hard data on whether cells are really undergoing division. And this uses stable isotope labeling. So he gives the mice N15, a stable isotope of the usual nitrogen 14 thymidine. That's incorporated into DNA. But the key thing is this is a quantitative measure. So he was able to distinguish between cells that had just repaired a bit of DNA and cells that were actually undergoing division. And so when I saw that paper, I approached Rich and said, gee, we should look at this in exercise to really try to figure out whether we're driving cardiomyogenesis. And in fact, we are. So we looked first in young adult animals, shown on the left there, and this was published in Nature Communications some years back. And if you just let the animals run, again, not 3 hours of swimming a day, but just let them run voluntarily to the extent that they want, although they do run a lot, there's almost a 5-fold increase in the rate of new cardiomyocytes being born over a 2-month period. And so that's a pretty dramatic increase, but I just want to hasten to point out that part of that is because the basal rate, the baseline rate of cardiomyogenesis, again, in the adult heart is quite low. And so what we saw in these mice, which has also been reported for people in the most reliable studies and is consistent with what Rich had seen previously, is about a 1% per year birth of new cardiomyocytes. So with exercise, that goes up to about 5%, 4.6%, in young adult mice. Interestingly, more recently, we looked in old mice, and you see a similar phenomenon in terms of the relative increase. Actually, the relative increase may be even slightly more, but the baseline rates are even lower. So what happens in old mice is that you increase the rate of birth of new cardiomyocytes sort of back to the baseline you see in young sedentary mice, but still that's, quite a dramatic increase. One of the questions that always comes up that we don't have a great answer for is how important is that, right? So the absolute number of cells that are being formed is relatively small, but there's a big relative increase. And I guess I would say in response to that, we don't know. It's hard to have clear data about that for reasons I'll expand on. But there are data, at least in animal models, that it takes a surprisingly small number of cardiomyocytes being lost over time to actually lead to a lethal dilated cardiomyopathy. So, for example, Rick Kitsis at Albert Einstein published some years ago in JCI that in constructing a mouse model where there was programmed cell death of cardiomyocytes that honestly was on the order of a handful of cells dying per 10,000 cardiomyocytes, over a period of just a few months, that would lead to a lethal dilated cardiomyopathy. So what we're talking about here is about an order of magnitude more than that. And so it's possible that that changes the balance between cell loss, cardiomyocyte loss, and cardiomyocyte birth in a meaningful way. The other thing that Rich pointed out is if you think about this and just do the back of the envelope calculation really through the magic of compound interest, if you take the baseline rate of 1% and increase that significantly, and he just did the calculation for 3%, and again, we're above that, we're closer to 5%, as individuals age, assuming this is consistent across the age of the individual, by the time someone gets to be 50 years old, 80% of their cardiomyocytes, this is again a calculation, not experimental data, would be younger, would have been born after the birth of that individual. So, that could be meaningful. But, I think, again, the thing I want to emphasize is that one of the challenges in sorting out exactly how meaningful that is that all the pathways that we've found that seem to regulate cardiomyogenesis or birth of new cardiomyocytes also have other effects. And so it's hard to design a truly clean experiment to say, is it really the addition of this small number of cardiomyocytes over time, or is it these other beneficial effects that affect things like cardiomyocyte survival and function? But conceptually, I think one of the really interesting things about this that it highlighted for me that I hadn't really thought much about before is the idea that organs are not necessarily all the same age, right? I mean, your bone marrow turns over much more rapidly, and so your bone marrow has much younger cells than you are. But even things like your heart or your brain or other organs could differ in their age and could differ biologically in their biological age than your chronological age. And that's something that we'll come back to at the end. So then, the question in a practical sense really becomes, what are the mechanisms, the specific pathways that regulate this response to exercise in all its benefits, including this cardiomyogenesis. And so we've spent a lot of time over the last decade or so trying to understand those pathways. And these are some of the ones that we've been interested in. But as a clinician, the practical question becomes, which of these pathways are potentially targetable? Which of them could we develop a medicine for that would manipulate and the pathway in a similar way that exercise does and potentially recapitulate some of the benefits of exercise? I do want to say at the outset that I don't think there'll ever be a medicine that recapitulates all of the benefits of exercise because they're just so protean and pleiotropic, but it's possible that we could at least exploit some of these pathways to mimic some of those benefits. And so what I'm going to do for the rest of the time is really talk about two examples from our work. And I've chosen these because I really think they are druggable targets. So there are things that could be moved forward, we hope, translationally. For one of them, there are actually FDA-approved drugs that actually mimic what we think we're trying to accomplish with this pathway. And as I mentioned, I'm trying to talk some of the companies behind those drugs into looking at this in the context of heart failure. And these two examples are also from two different classes of molecules that I think illustrate how the scope of things that we think about targeting therapeutically has really expanded in recent years. So it used to be that you could only think about, GPCR targeting or kinase inhibitors or things like that. And now I think the universe of possibilities is much broader. So again, an exciting time to be in biomedicine. So the first example I'm going to talk about is a non-coding RNA that we call link exact 1, which we think regulates both growth of the heart and cardiomyogenesis. And it's something that we'd like to inhibit. And the second one is a series of secreted molecules that are proteins that circulate in your blood. And that's the one where there are already FDA-approved drugs that target this. I want to mention at the outset that each of these projects were led by incredibly talented young scientists who were in the lab but have recently launched their own laboratories. So the non-coding RNA work was done by Pauley, who just recently started his own lab in the anesthesiology department at Mass General. And then the activin receptor ligand work was led by Jason Rowe, who started his lab in the cardiology department at Mass General as well. And I just want to highlight what an amazing job each of them has done. So first, thinking about the non-coding RNAs, I think everyone here knows that, what we learned in medical school, DNA makes RNA makes protein is true. That is the central dogma of molecular biology, but it's only true for a very small fraction of the DNA in our bodies. A lot of the DNA in our bodies is, in fact, transcribed into RNA, but that RNA never makes protein. So what do those RNA molecules do? A lot of them regulate expression of other genes. And what's interesting to me about that is, first, we're still learning lots about how this works, but also it's possible to target those RNAs with a variety of approaches, including antisense reagents. And as you know, there are antisense reagents on the market that target things like cholesterol metabolism. So it's feasible to think about targeting in that way. So a number of years ago, Habo in the lab said, well, why don't we look at the long non-coding RNAs? There are long and short and different classes that are altered by exercise. And so he identified 25 long non-coding RNAs, 11 that decreased with exercise in the heart, and 14 that increased in exercise in the heart. And then he compared those to the long non-coding RNAs that changed in pathologic models. So TAC is that transverse aortic constriction model that in some simple way mimics the increased afterload you see in aortic stenosis. And he either looked at aortic constriction that led to hypertrophy, but before the hearts were failing, or a later date when the hearts were actually failing. And so first thing I want to point out is that, consistent with what I told you at the outset, we see this theme repeated that most of the things that are changed in heart failure or hypertrophy, pathologic hypertrophy, or exercise-induced hypertrophy are different, right? There's very little overlap. There are really only 6 that overlap that are changed in both the physiologic and the pathologic models. One of those 6 didn't replicate, so 5 replicated, and just showing 3 of them over here. Interestingly, for all 3 of them, again, as I mentioned before, they all change in opposite directions. So if you look at link exact 2, for example, here, in running, it goes up, but in the pathologic hypertrophy model, it goes down. Similarly for link exact 3, we were particularly interested in link exact 1, and I'll tell you why. So in running, this goes down, it decreases in the run hearts. And then in both of the pathologic models, both the hypertrophy and the heart failure model, it goes up. And so that was interesting, partly because, as I mentioned, there are drugs that inhibit RNAs that are FDA approved, about a dozen of them. And it's much more feasible to think about inhibiting a target like a long non-coding RNA than it is to think about, how you would enhance it or increase it, which could be complicated, but might involve, gene therapy or something like that. So we were particularly interested in this. It was the only one that was decreased in exercise, and almost always the desired change is mimicking what you see in exercise. So that was our hypothesis going into it. The other reason we were particularly interested in this is that some of you may know, long non-coding RNAs are not always well conserved through evolution. So it's possible that you would find something in a mouse heart that doesn't have a homolog, or a clear homolog in a human heart. And so one of the things that was intriguing about link exact 1 was that it was about 70% identical between humans and mouse at the sequence level. So that doesn't prove anything, but when something is conserved through evolution, it suggests that it's functionally important, but it also more importantly suggests that if we find something for the mouse, it might be translatable to people. And then in collaboration with Mike Bristow and then Soumya Das, Mike Bristow at Colorado and Soumya Das at MGH, we looked at link exact 1, the human homolog in human heart failure. And so in human cardiac tissue, these are patients with idiopathic dilated cardiomyopathy compared to control hearts. And then some of it circulates in the plasma. And so in human plasma from patients with heart failure compared with patients with arrhythmia and no heart failure, you can see that it increases as it increases in the hearts of mice with heart failure. So again, this doesn't prove anything, but suggested that here's something that decreases with exercise. So we thought that might be the beneficial pathway. And that would be feasible as a medical therapeutic target. And also it's closely conserved in the human situation. And it similarly is dynamically regulated in human heart failure, all of which was encouraging. So I'm going to skip over a bunch of studies that Habo did in vitro in cells to try to explore this, all of which were encouraging and led us to do in vivo experiments in mice. And so this is an example of one of those. So he injected a virus that carries a cardiac specific promoter driving expression of this long non-coding RNA, link exact 1. I should say it was on the prior slide, but Habo came up with this name. It stands for long non-coding exercise associated cardiac transcript link exact 1, that because none of these had been annotated previously. And so you can see that by injecting this virus in the tail vein, you get a cardiac, an increase in heart expression that's, somewhere in the, 5 to 8-fold range. And that's about what we see in both human and animal models of heart failure. So it's important in all these kinds of animal models to make sure that what you're looking at is pathophysiologically relevant. This is in the range that we typically see. And so just overexpressing link exact 1 is enough to induce pathologic hypertrophy. The hearts get bigger, the relative wall thickness measured by echo gets bigger, cardiomyocyte size gets bigger, you can see here, but there's no increase in markers of cardiomyocyte proliferation. And actually fractional shortening, which is a similar measure to ejection fraction, actually increases, as does ejection fraction. And markers of pathology like natriuretic peptides, ANP and BNP, which we obviously use clinically, increase quite substantially, suggesting this overexpressing this long non-coding RNA is sufficient to cause pathologic hypertrophy and a little bit of dysfunction without driving proliferation. The surprise was what happened when he inhibited it. So again, he's inhibited it a couple of different ways, but one way is using an antisense modified oligonucleotide similar to some FDA drugs that are already approved, but not for this target or indication. And so here again, he's getting about a 2-fold reduction in the expression in the heart with systemic delivery of this antisense reagent, which is again about the change we see in exercise. So in some ways it mimics what we see in exercise. And the surprise was that just inhibiting link exact 1 also made the hearts grow. So we're more typically used to the idea that a pathway, when we enhance it, makes the hearts grow, and then if we inhibit it, it makes them shrink. Here, if we enhance it or inhibit it, the hearts grow either way. But what's interesting is, so you can see the heart weight increases, the relative wall thickness increases, the cardiomyocyte size increases. But here, there are no markers of pathology. So natriuretic peptides and myosin heavy chain and so on actually look like a more physiologic kind of growth. And this transcriptional coactivator, PGC1, actually increases a hallmark of physiologic growth. And importantly, the systolic function of the hearts actually improves. So remember last time, if we overexpress it, it gets worse. Here, it actually improves. Quite different here, all of the markers that we've looked at, EDU incorporation, Ki67, phosphohistone H3, suggest an increase in cardiomyocyte proliferation. And remember, exercise drives an increase in cardiomyocyte proliferation. This suggests that this pathway, link exact 1, may be contributing that when it decreases in exercise. I would hasten to add, these are just markers. with the caveats that I mentioned earlier, and we haven't done the stable isotope labeling in these yet. Well, so exercise also protects against pathologic stress, and in many ways, in many settings, can rescue it. So we looked in several models of pathology, the aortic constriction model that I've already mentioned. This is a myosin heavy chain mutation that was discovered by the Seidman laboratory that causes a dilated cardiomyopathy, and they developed a mouse model of this. And then this is ischemic injury. And in all these cases, if we inhibit link exact 1, again, with this antisense reagent, we can decrease it in the heart and drive a pretty substantial improvement in systolic function. And I just want to hasten that, particularly in these cases, we're delivering this, in this case, to animals that are quite old and already have a dilated cardiomyopathy. And in this case, after the ischemic injury and reperfusion. So we're delivering it in a clinically relevant time frame, not delivering it beforehand and then subjecting them to stress. And in addition to the functional improvement, the other dramatic thing is that fibrosis, the amount of scar at the end of the experiment, in each case dramatically improves, about half what it was before. And so we think that's a combination of effects both on cardiomyocytes and on fibroblasts, which I can expand on more if people are interested in. And in each case, these is associated with an increase in markers of proliferation. So, again, recapitulating this idea that things that are changed in exercise, when we mimic that change in exercise, we often recapitulate the benefits you might see in a pathologic setting as well. And so we find this target really interesting. It seems to act as kind of a toggle switch between a pathologic growth of the heart and a physiologic growth. So when it goes up in these disease models or in human disease, that alone is sufficient to induce pathologic hypertrophy, suggesting it's playing an important role. And when it goes down, that alone is sufficient to induce physiologic hypertrophy, but also protect against heart failure. In the interest of time, I'm not going to go into the mechanism because this was published last year, but we think it works through 2 ways. One is binding this microRNA that we had previously identified as important in the exercise response, miR-222, and then also by regulating transcription of its nearest genomic neighbor. And that's probably the pathway that drives proliferation. So we're excited about this because it can be effectively inhibited in vivo, at least in animal models. And we've recently gotten a Harrington Discovery Institute award to actually explore the translational implications of this and see if we can develop this further as a target for heart disease. The other pathway that I wanted to talk about, again, I think is even a little closer to the clinic because there are drugs available that inhibit this pathway. And this is a group of secreted proteins that I class as activin receptor ligands that regulate cardiomyocyte hypertrophy or growth, inflammation, and metabolism. And so just to say a little bit more about these, I think the most famous member of this pathway, this is a branch of the TGF-beta family, is something called myostatin that was discovered by Si-Jin Lee at Hopkins some years ago, also known as GDF8. But there's a group of these very closely related proteins, GDF8, 11, 15, and activin, which is encoded by inhibin A. And they're best known for being regulators of skeletal muscle growth. And so, for example, for myostatin, when Si-Jin Lee knocked this out in mice, you can see that the skeletal muscle is often 2 or 3 times the normal size of skeletal muscle. So a major regulator of skeletal muscle growth and metabolism. There are naturally occurring mutations in these pathways in every mammalian species that people have studied, including man, shown here. So there are rare families with myostatin mutations. In these cattle, when they have myostatin mutations, they have enormous skeletal muscle. These are Belgian blue cattle, and that's why they have such big skeletal muscle. When they're homozygous, they have to actually be delivered by cesarean section because their muscles are so large. And then all of these ligands bind a common set of receptors, the type 1, and shown here the type 2 receptors that then regulate AKT signaling and SMAD, phosphorylation, and nuclear translocation. And in response to that, they induce the secretion of something called follistatin-like 3, which can then be used as a biomarker for activation of the pathway. So one of the interesting and challenging things is that there are so many different ligands, and there are some endogenous inhibitors that I'm not showing here. it's a little hard just from measuring those ligands to know what the state of this pathway is, but follistatin-like 3 can serve as a useful biomarker because it's induced by all the ligands. And so if the net effect of an intervention is to increase signaling through this pathway, we see an increase in follistatin-like 3. So we became interested in this some years ago when, in collaboration with Rob Gersten over at Beth Israel Deaconess, we looked at circulating proteins. So this is a proteomic study, targeted proteomics of circulating proteins in people in the Framingham Heart Study as they age. And so follistatin-like 3 emerged as one of the top proteins that correlated with age. And you can see that this monotonically increases as the population ages. And of the ligands that were present on the platform at that time, the only one that positively correlated with that was activin itself. So this suggested that in people as they age, activin was increasing and it was driving this biomarker increase in follistatin-like 3. And this is just some of the other data. Now, the Framingham Heart Study, as you all know, is a general population study. So there's not a lot of prevalent heart failure. But then we looked similarly in a group of aortic stenosis patients where there was much more heart failure. And interestingly, once again, not only does follistatin-like 3 correlate with age, shown here, but it also correlates with heart failure severity. So New York Heart Association or NT-BNP levels correlate pretty significantly and positively with follistatin-like 3 and with activin levels. So we were particularly interested in this, partly because one of Jason's interests is aging biology and how that contributes to heart failure. And both activin and follistatin-like 3 are what are called SASP proteins, senescence-associated secretory phenotype proteins, meaning that as cells undergo this senescence phenotype as they age or are stressed, they release a series of secreted proteins, including activin and follistatin-like 3 in many instances, that have important effects on immune cell function, on inflammation more generally, on tissue remodeling, and can induce senescence in other cells. So this suggested that there might be some really interesting senescence biology connection here. This is a famous Nature paper. These two mice that look so different are actually litter mates. So they were born at the same time. But in this mouse that looks young and healthy, the senescence cells were genetically removed in this mouse model, and in this mouse they weren't. And you can see how removing those senescence cells has a dramatic effect on at least the overall appearance and some of the health characteristics of these mice. And so people have been quite interested in what are called senolytics, medicines that might actually inhibit senescent cells or remove them from the body. So the other reason we thought this was interesting is, as I mentioned, there are a bunch of companies that have developed inhibitors of this pathway. Most of those inhibitors have been developed to either target metabolic disease or skeletal muscle disease like sarcopenia, or in some cases muscular dystrophy, or primary pulmonary hypertension, where this pathway through the BMP side of this pathway contributes at least to genetic disease. And again, two of these drugs that are inhibitors have recently been FDA approved. So, the question at the time was, is there a role for these activin ligands in heart failure? Do these activin ligands not only regulate skeletal muscle, but perhaps another form of striated muscle, the heart? And so I know we have some fellows and trainees on the Zoom call. So, I just think it's worth pausing and emphasizing that anytime you have an idea like this, no matter how much you think it's a novel idea and no one has thought of it, it's always important to go and kind of humble yourself before the literature, because almost anything you've thought of, someone else has thought of. And so at the time, I said to Mike Marset, who was the fellow working on this in the lab at the time, we should go and look and see what's known about these ligands in the heart, and whether there's something there. And so I think we had a little bit of a generational divide when I said go and see what you can find out. I think for my generation, I was thinking library, and I think Mike was thinking internet. And so when he went and looked, he found a number of authoritative sources on the internet, including something called getbuff.com. and something called MuscleDawg, DAWG.com. I'm from New York originally, so I can translate that. And interestingly, both of these were selling inhibitors of this pathway with the idea, as I showed you in the myostatin knockout mice, that you could grow your skeletal muscle. So this was for bodybuilders. First of all, this is not an off-label recommendation. These are oral pills that I suspect get chewed up in the stomach and don't actually do anything, so don't waste your money. But interestingly, in the FAQs from MuscleDawg, they actually had the question, will these inhibitors cause heart muscle or smooth muscle to grow? And I've got to say, I was just so impressed that MuscleDawg had the insight to actually recognize these were muscle lineages and, this might be a concern, and put it in their FAQs. But even more impressive than that, MuscleDawg actually had the answer. And so they quite definitively said, no, myostatin is quite specific for skeletal muscle, so these inhibitors won't do anything to heart or smooth muscle. So I said to Mike, well, we've got the answer. Why don't we just stop the project right there? MuscleDawg says it doesn't, so that must be right. But fortunately, Mike was a bit more persistent. And so over the years, both from Mike Morissette and Pablo Quintero and Jason Rowe, we've moved this project forward. And once again, the theme is I was wrong. MuscleDawg was wrong. It does seem to have an important effect in the heart. And so how do we know that? Well, so the first thing is to go back to those models where we can say, what if we take a normal mouse that's healthy, no comorbidities, and just overexpress activin itself and kind of mimic what happens with aging or other settings where activin is increased. And just doing that is sufficient to induce cardiac, shown with the echoes in the middle, and cardiomyocyte dysfunction. So these are isolated cardiomyocytes. That's a normal one that got the control virus, and this is one in which activin is being overexpressed. And it's hard to tell, but it is actually moving. It's just moving much less. And so both movement of the cardiomyocytes and calcium handling are dramatically impaired. So activin is sufficient to cause cardiac dysfunction and in some cases heart failure. The more clinically relevant question is, what if we inhibit it? And so these are published data, so I'm just going to go quickly through, but in 3 different models, that aortic constriction model, that dilated genetic cardiomyopathy model, and then an age-related model of heart failure with preserved ejection fraction, in each of these models, inhibiting activin, even in a clinically relevant time frame after the animals have heart failure, is sufficient to substantially rescue function. So you can see here, aortic constriction, the animals develop dysfunction, and then we start treating them, and pretty quickly their systolic function improves. In the heart failure with preserved ejection fraction, systolic function is normal, but at baseline, so hard to see changes in that, but things like strain improve, and exercise capacity pretty dramatically improves. And we've done this both with genetic models and with inhibitors. There are, as I mentioned, multiple inhibitors out there. These data are an antibody to the receptors, but they're also soluble receptor ligands. I won't dwell on mechanism, but it does have direct effects on cardiomyocytes, regulating calcium handling through affecting the stability of this key protein SERCA2a that regulates calcium uptake into the sarcoplasmic reticulum. But it does other things and quite dramatically reduces the amount of inflammation that we see in the hearts in these heart failure models, particularly with myeloid cells. So neutrophils and monocytes are, you can see almost reduced to zero, quite dramatically reduced by inhibiting this pathway. So, again, we're intrigued by this. And these studies were done in the context of age-related heart failure. But as we've studied this pathway over the subsequent years, what we have found is that it actually seems to have important effects beyond just age. And I want to come back to that idea that age, although we think of it as a constant, inexorable variable, so we don't even talk about it most of the time, it's not quite so constant, right? So biological aging varies, and this is the some famous examples that people have observed over the years, that when people become president, their rate of aging, understandably because of the stress that they're subjected to, changes quite dramatically. And as I mentioned earlier, the notion that exercise could drive some regenerative capacity in the heart suggests that the age of those cells and the age of cells in different organs may vary. So I want to give one example of several that we found recently where surprisingly this pathway is relevant not just to old people but to heart failure and other settings. And this example relates to peripartum cardiomyopathy. So as all of you, I think, know, this is an idiopathic form of heart failure that's thought to exist on a continuum with preeclampsia. It's relatively rare in the United States, about 1 in 1,000, but in other parts of the world, including Nigeria, it's astoundingly high reported incidence. And interestingly, women who have either peripartum cardiomyopathy or preeclampsia are at higher risk for cardiovascular disease throughout their lives subsequently for reasons that aren't well understood. So a number of years ago, Jason led in the lab another proteomic study to say, if we look at the proteins that are circulating in the plasma of women with peripartum cardiomyopathy compared to maternal age and gestational age matched controls, what do we see, right? Is there any clues to what the etiology may be? Because there's long been this sense there may be circulating factors, and soluble FLT1 was one that was identified many years ago that contributed to this condition. And so really surprisingly, when he did that, in both peripartum cardiomyopathy and preeclampsia, the top pathway of plasma proteins that came up in both of these conditions was that SASP, or senescence-associated secretory phenotype proteins, SASP proteins. So you can see it's quite dramatically increased, way more than any of the other pathways. And so although women with PPCM or preeclampsia are slightly older than the average pregnant woman, these are young women. The average ages were 32 and 34 in these two different cohorts. And so we were initially scratching our heads and saying, why is it that we would see this senescence, aging-related pathway as the number one thing in both of these conditions? And then as we thought about it, we realized that the placenta is born and destined to die 10 months later. So the placenta essentially has to go through a process of involution and accelerated aging so that you don't bleed to death, when the baby is born. And so then we looked at placentas, and this is work done by Jason and Claire Castro, a postdoc who's been working with Jason. We found that the 28 common proteins that were in the plasma of women with PPCM and preeclampsia were all, or mostly increased in the placentas of women with preeclampsia. You can see here that the number one upregulated protein at the gene expression level was activin A, which is encoded by the inhibin A gene. So that's number one increased. And number two was follistatin-like 3, again, suggesting that the increase in this pathway is overall increasing signaling through activin. All right, so does this relate to the clinical presentation? And so just to quickly go through this, activin A, this is all unpublished data, but hopefully soon will be. Activin A correlates with systolic dysfunction, both in preeclampsia and peripartum cardiomyopathy, and New York Heart Association and NT-BNP levels. So then functionally, what does this mean? And so there are 1 or 2 models that are reasonable of peripartum cardiomyopathy. And so Zoltarani developed one published in Nature some years ago. It's a PGC1 cardiomyocyte-specific knockout, and those mice develop progressive cardiomyopathy with successive pregnancies. And so we used that model and tried 2 different interventions. The first was using those senolytics. So something called fisetin is a senolytic that removes senescent cells. And you can see that in the mice that are developing this peripartum cardiomyopathy with pregnancy, when you treat them with fisetin, you get actually a pretty substantial increase in cardiac function. And the other approach that we used was inhibiting activin. Activin is important to development, so you can't do that during pregnancy. But in these studies, this was done in postpartum mice. And just to emphasize, quite a few of the women with peripartum cardiomyopathy present shortly after delivery. And here again, you can see there's a pretty dramatic increase in cardiac function in these mice that had multiple pregnancies and were more severely affected. So just to wrap up and summarize, I think what we've talked about is that the heart grows in response to both physiologic and pathologic stimuli, but they have very different outcomes and very different underlying pathways when we look at a kind of molecular mechanistic levels. And those pathways are enriched for therapeutic targets. So we think that exercise is a fruitful area to sort of think about when looking for new therapeutic targets. we think of aging as a constant, but I think our studies and many others suggest that biological aging, which is distinct from chronological aging, although closely related, can vary not only among individuals, but across organs, and can be modified by environmental stimuli and different manipulations. So worth thinking about in this context, since it's so closely linked to heart failure. And we talked about two potential new targets that are exercise regulated and druggable. I think I glossed over and didn't mention, but activins regulate skeletal muscle growth. And one of the ways you respond to exercise to increase your skeletal muscle size is by inhibiting this. So once again, in mimicking the change we see in exercise has the beneficial effect. But the first one we talked about is this non-coding RNA that we can effectively target in vivo with antisense, at least in animal models. And we're trying to explore the translational implications. And the second is this activin signaling pathway that is part of senescence, but probably is important not just in chronological aging, but in other settings where stress induces biological aging, or at least an induction of these senescence pathways. I don't have time to talk about it today, but as Dr. Prabhu mentioned, I recently moved to Michigan to lead a heart and brain institute, and each of these pathways seem to have important roles in the brain as well, and that's the kind of thing that has gotten us interested in this. And just in the last minute or two, I just wanted to have some closing thoughts that are perhaps a little closer to humans. The most common question I get asked is, so you're saying exercise is a medicine. When can I take a pill and I don't have to exercise? And just to emphasize, that's not the goal. And I don't think personally that we'll ever come up with exercise in a pill, although this is another thing that's sold on the internet. It's exercise without an E, so don't trust it. I think for those of us who can exercise, we should probably not take the escalator up to the gym. And sometimes our brains are a little bit too smart for us and outsmart the potential benefits. As a clinician, certainly my philosophy is more reflected here. Everyone's too busy, but would you rather exercise an hour a day or be dead 24 hours a day? I think it does have important effects on an outcome. So I think it's particularly hard for those of us who, as we progressively become more sedentary and wind up at jobs where we're at a desk or at a computer much of the time. I do have a treadmill desk, but I have not been able to or gone quite this far with it. Some of what people have advocated is probably also not a good idea. I think that there's something about electricity and water that might not make this good for your health. The other things that I get asked often are, well, how much do I need to exercise to get these benefits? And of course, like everything, it depends what your goals are. If you're training for an Ironman competition, that's one thing. But for most of us who want the health benefits, it takes surprisingly little. The biggest benefit we see is an association from comparing people who are sedentary to people who do modest levels of activity. And this got a fair bit of attention because based on observational data, it was estimated that just 10 minutes a day of walking, so modest activity, was associated with dramatically improved clinical outcomes. And then people say, well, what exercise should I do? And again, it depends what your goals are, but I think it also depends what you enjoy and what you will do. And so if you're a fish out of water on a treadmill, it's better to find something else that you'll stick with and do consistently. And then the other really interesting question I get asked is, why is exercise different? Cardiologists in particular say, well, gee, if I have a heart attack, isn't the surviving part of my heart just exercising? It's working harder. Why does that lead to adverse remodeling in many instances? But exercise, you're telling me, has benefits. And of course, I don't really know the answer to that. I would say in a teleological sense, there's a strong evolutionary pressure to select for a beneficial response to exercise in our ancestors, and nothing driving selective pressure on things that like heart failure or heart attacks that probably don't affect reproductive success. I think there are differences. My friends who are true physiologists point out there are differences between the mechanical forces, but my hypothesis is it's not all hemodynamics, that there are circulating factors that bind receptors and change the response. Some of those circulating factors we've talked about, and we're particularly interested in the idea that exercise seems to drive a regenerative response in the heart. We didn't really talk about this, but there's a lot of literature previously from people like Rusty Gage about how it does a similar thing in the brain. And so are these independent things, or are there signals that are circulating that are contributing to that? And that's something we're quite interested in. And finally, just to highlight again, the people who really did the work, Habo Lee and Jason Rowe really led these studies, and Claire Castro not shown here. And then I've moved to Michigan. This is the group in Michigan very grateful for the founders and our collaborators, and delighted to answer any questions if there are.
Thank you for that phenomenal talk, sir. Really appreciate that. We have one question in the chat from Dr. Fadi. Excellent talk. Thank you. Since the benefits of exercise are pleiotropic, would targeting multiple pathways via polypharmacy be feasible in those who are unable to exercise? Most studies focus on one pathway at a time. Any insights for designs of future studies and trials, keeping in mind the benefits of exercise on other organs besides the heart that may contribute to the heart disease?
Yeah. Oh, thanks so much, Fadi, for that wonderful question. I think it's a great point. I guess I feel like we're still relatively early in sorting this out. And so I'm not sure that I'm at the point of saying, here's the 3 drugs that we'd want to use. And we're still trying to kind of validate and explore, certainly in a human context, the individual drugs. The combinations get more complicated. And then I would say that they are pleiotropic, but as mentioned briefly, one of the interesting things is that all of the pathways we find themselves are pleiotropic, right? And so activin does a bunch of things, and I just kind of briefly alluded to them, but it changes growth, it changes metabolism, it changes inflammation, it does a bunch of these, and it changes calcium handling in cardiomyocytes. And so, although, we sort of often have this somewhat reductionist view that we're going to manipulate this one pathway and it has a direct route that has some beneficial effects. I think we often see that these interventions actually affect a network of pathways and gene expression. And so again, we're still trying to fully understand that. But it does suggest that at least with the things we found in exercise, they're doing so many things that there's at least some hope that the pathways that we see that may affect cardiomyocyte proliferation, may affect cardiomyocyte survival, may affect fibrosis, also affect other relevant phenomenon and pathways and biology that may be beneficial. But as a basic scientist, I would say we would just be thrilled to see some of these things get to the point of being tested in man, which is obviously the next. First, are they safe, and then secondly, can you test them? For the activin family, what's interesting is there are safe drugs that are FDA approved for other indications. And so again, we would love to see a test of whether you can see anything similar to what we see in mice in people using those. And then, I think your important point is not only polypharmacy, but also, any treatment would have to show benefit on top of standard of care, right? It's not enough just to say by itself, this is good. In a human context, we'd want to show that it actually improves overall outcomes, beyond that. And that's a high bar. It's a particularly a high bar for HFrEF, where there are so many treatments, at least, 5 or 6, that we always use in those patients. For HFpEF, it's a little bit lower bar because we don't have that many things that are really shown to be beneficial. But yeah, thanks so much for the question.
One of our fellows, Dr. Patel, is saying that she's going to go to the gym after listening to this talk. So you have left an impact on us fellows. A question I had, depending on different heart rate exertions, do you think different pathways get activated? If somebody is, my Apple Watch keeps a track of it. My zone of heart rates are sometimes in the zone 5, heart rates above 160, sometimes like 140s to 160s. Do you think at different heart rates or different exertional levels, different pathways are activated that are beneficial?
Yeah, for sure. No, great question. Well, and first of all, just to respond to Dr. Patel, that's great. Like, I think if I accomplish nothing else, if I nudge people to do a little bit more exercise, I think that's probably the most important thing, especially for hopefully young, healthy people, just to maintain that and incorporate that into your lives is probably the most important message. I think in terms there certainly are differences in the response to both different forms of exercise, and in mice there's a limited repertoire, right? So it's hard to get mice to lift weights. We've tried, but mostly, although people do have models where they attach weights to their tails and make them climb things, that just seems cruel. But is an attempt to get at resistance training. So most of the mouse studies are really done in some form of kind of endurance exercise, or endurance exercise training, although again, you can change the intensity. And so things like wheel running or kind of low intensity loads because there's ideally almost no resistance to the wheels, but then we can work, exercise them on a treadmill, which we also do with different different inclines and so on. In humans, actually, Sawala Gusa, when he was in the lab a couple of years ago, actually looked at young, healthy subjects who were either asked to kind of do a slow jog for a certain set period for 5 miles or something like that, or to run as fast as they could, and then did a similar soma logic targeted proteomic approach to say what changes. And yes, there are some common things, but there are pretty dramatic differences as well, which I think is really interesting. So I'm sure you're right, and so that paper was published in Scientific Reports, but you can look at it. I mean, there are we haven't checked the functional roles of those proteins, but for example, in the high intensity group, there were a number of proteins modulated that have been linked to atrial fibrillation in other contexts. And we know that, my kids often accuse me of trying to suggest exercise cures everything. I don't think it actually cures everything, but it's pretty close at preventing most bad things. But the one exception to that, or one of the few exceptions to that, is, as you all know, we do see atrial fibrillation, particularly in high volume, high intensity athletes who are otherwise healthy. And you can recapitulate some of that in animal models. So we thought it was interesting that in young, healthy subjects who more intensely ran, and then we looked at their acute proteomic changes compared to those who just kind of did a slow jog, we saw, a handful of proteins that had previously been linked in genetic studies and other work to atrial fibrillation. So I'm sure there are differences. And then the classic thing, not from our work, but from other people's, is when you exercise intensely, people have documented an increase in cortisol levels, whereas if you exercise modestly, there's usually a decrease in cortisol levels. And similarly, glucose may be related to the cortisol, but sometimes intense exercise is associated with a somewhat paradoxical increase in glucose because the sympathetic tone and the cortisol all mobilize glucose. Whereas if you do more moderate levels of activity, generally your glucose decreases with that. So I'm sure there are differences, and it's just so hard. There aren't great studies, in humans really teasing out what we'd all be interested in is which of those really have beneficial effects. Although again, I would argue that if you look at the observational data, the biggest difference in outcome is associated, all observational data, when you compare modest levels of activity to sedentary people. And then the additional benefit as people go on to the much larger scale activity is less impressive. It's a kind of a flatter slope as you go beyond that. And I mean, my friends who are, committed and accomplished athletes will say, well, there aren't that many people who are out there who are running, 40 miles a week or something like that. So we don't know. But again, my read of most of the data, perhaps a rationalization because I'm not an accomplished athlete, is that, probably most of the cardiovascular benefits and health benefits occur at relatively modest levels, but it's hard to know for sure.
That is fascinating. Thank you for answering that. I believe we are towards the end of our time. Again, thank you, sir, for giving us this phenomenal lecture. We had a great time, learned a lot from this. And for CME credits, please text Shannon and we'll go from there.
Great. Thanks so much. Thanks, everybody. Appreciate the interest and the opportunity. Have a great one.